Gregor Mendel and his peas. Between the post-Neolithic revolution of around 10,000 years ago and the mid-1800s, there was a lot of progress in agriculture, such as the invention of the plough, the discovery of fertiliser and the use of irrigation, but not much progress in the science of plant breeding. This all changed when Austrian monk Gregor Mendel conducted a series of experiments on 30,000 pea plants at his monastery around 1860. These experiments helped him to understand the basic principles of genetics. In one experiment, he cross-pollinated plants with green peas and plants with yellow peas. He then examined the offspring to see what the results were. The offspring of this cross-pollination, called the first filial generation, or F1, all had yellow seeds. This showed that the trait for yellow seeds was dominant over the trait for green seeds. Mendel then allowed the F1 plants to self-pollinate. The offspring of this self-pollination, called the second filial generation, or F2, were a mix of yellow and green seeds. This showed that the trait for green seeds was still present in all yellow F1 plants, even though it didn't show in that generation. Mendel's experiments proved that traits are passed down from parents to offspring in a predictable way. He also showed that there are two types of factors, called genes, that control each trait. One version of the gene, called an allele, may be dominant and is always expressed, while the other allele may be recessive and is only expressed if the plant has two copies of the same allele. This knowledge is still being used today, allowing plant breeders to use DNA-based markers to select for favourable alleles that result in new varieties of plants with improved characteristics.